Let us start with the today's episode. Now, what are the main learning objectives of today's lesson? First, we will discuss about the digestion in small intestine. We will discuss about what are enzymes. We will discuss the action of bile, the action of pancreatic juice, the action of intestinal juice, and the digestion in the large intestine also. If some digestion happens in large intestine, we should discuss about that. So let us see what is there in store for us. Now, first, before starting this lesson on digestive system, we should know about what are enzymes. Now, in chemistry, I think you have already studied about what are catalysts. Now, here we will discuss about enzymes which are biocatalysts. That is, as they are being used in the biochemical reactions of the body, they are called biocatalysts, and they are mainly made up of proteins. What they do, they speed up the rate of most of the chemical reactions in our body, and there are many enzymes which are concerned with different types of processes which are happening in our body. Like we have given just two examples: digestion, respiration, etc. Many enzymes are involved in them. Now, enzymes, they act on only one type of substance. If any enzyme is acting on protein, it will be acting on the substances which are made up of proteins only. If any enzyme is acting on starch, it will be acting on starch only. That is, it acts on only one type of substance. So, an enzyme which is acting on starch will not also act on protein, because it is an enzyme which is acting always on starch. Now, next, like catalyst, we can say we can use enzyme again and again. So, once it is used, it can be reused again. It is not wasted, it is not destroyed, so it can be used again and again. But this enzyme always acts on a particular pH value. The optimum temperature is 30 to 40 degrees centigrade, and after that, we can say it becomes inactive. So the optimum temperature is 30 to 40 degrees, and it becomes inactive and denatured after that. So this is what the enzymes are all about. So we have discussed about what are enzymes and how are they acting on one type of substance on a certain pH, and they are being used again and again in certain chemical reactions to speed up the reaction and to change the rate of the reaction. Now let us see how does they act in the digestion in small intestine. Now first, in the small intestine, what is done, the food is digested as well as the food is absorbed in the small intestine. Now the digestion and absorption, as both are taking in the small intestine, there are some substances which are being used in it. So what are that? The food here mixes with the bile juice, with the pancreatic juice, and with the intestinal juice. Now all the above three juices, they are all alkaline in nature, and the acidic chyme, so the food which has been already digested in the stomach with the help of HCl, it is slowly passing on to the small intestine via the duodenum. So that food is already acidic in nature, but that is being neutralized and converted into alkaline medium, and the pepsin is inactivated. The pepsin already present in the food chyme is inactivated in the small intestine. So what is happening here? Now let us see the action of bile. Now what is bile? Bile is actually a yellowish colored. I've already highlighted with blue color. You see, it is a yellowish green color watery fluid. So it is a yellowish green color watery fluid which is actually produced in the liver, and it is transported through the hepatic duct. And why the color of bile is such? The color of bile is such due to the presence of two pigments, bilirubin and biliverdin. And how are they formed? They are due to the breakdown of certain dead and worn out red blood cells. Now here first mention should be made that bile does not contain any enzyme. Bile contains only bile pigments and bile salts. Follow them, understand them, and try to learn them. That bile does not contain any enzyme, they contain only bile pigments and bile salts. And that sodium carbonate is present in bile, which neutralizes. What does it do? It neutralizes the acidic food or the chyme into the alkaline chyme. Okay, is it clear? So what is present here? We can say that the acid content of this food is neutralized by it is sodium bicarbonate. It is sodium bicarbonate is present. So it is sodium bicarbonate is present, which converts the acidic chyme to alkaline chyme. So fat is also converted to emulsified fat, and the emulsification thing is done in this small intestine with the help of the bile juice. So is it clear to everyone? See, this is the function of bile. Just go through it once. I think you will understand, as I have mentioned each and everything in bullets, in points. So once you go through it, that sodium bicarbonate is present, bile salts and bile pigments are present. It converts the fat to emulsified fat, that is, emulsification happens, and it facilitates further the digestion of fat, and the acidic chyme is converted into the alkaline chyme with the help of the sodium bicarbonate. Okay, so next we will move on to the action of the pancreatic juice. Now, first, when we are studying this, the pancreatic juice is produced by the pancreas. The pancreatic duct, what does they do? These duct pour their secretion in the duodenum. They are pouring the secretion into the duodenum, and there are 4 types of enzymes containing in the pancreatic juice, namely trypsin, chymotrypsin, amylopsin, and steepsin. So these are the main 4 enzymes. Now from the first till the fourth, we will be studying a little bit in details. We should know about them, right? So as we have already told that they have enzymes have a similar, they act on similar kind of substances. One enzyme can act on one type of substance, and they are acting on same 30 to 40 degree temperature, and they can be used again and again. So how these 4 enzymes are acting, we should know that. So let us see. Now first, when we are taking trypsin is a pancreatic protease, right? It is a pancreatic protease, and it is formed by the action of enzyme from trypsinogen by the action of an enzyme called enterokinase, or it is also called enteropeptidase by the inner lining of the duodenum. Okay, by the inner lining of the duodenum. And it is acting on mainly proteins. So it is acting on proteins, proteoses and peptones. So it is acting on proteins, proteoses and peptones. They all in the presence of this trypsin, what they are doing, they are all converted into polypeptides. So these all are converted to polypeptides in the presence of this trypsin. So this trypsin is helping them to convert this pepsin, proteoses and proteins to the polypeptide, but the medium should always be alkaline. So the medium is always alkaline. The medium is alkaline. So this enzyme is a pancreatic protease, means which acts on proteins only, proteins, proteoses and peptones, and is converted to polypeptides. So this is the function of this trypsin enzyme, that is pancreatic protease. Now comes the chymotrypsin. So it is an activated form of enzyme formed by the action of trypsin on chymotrypsinogen. So chymotrypsin, on the action of chymotrypsinogen, inactive form is converted to chymotrypsin. So how this is acting? This is also, we can say that, acting on specific proteins and is converted in the same way to polypeptides in the alkaline medium. Alkaline medium, chymotrypsin converts the specific proteins to polypeptides. Next comes the amylopsin. It is a pancreatic amylase. There is a certain difference. Previous two were pancreatic proteases acting on protein enzymes. But this is an amylase which acts on starch and complex sugar and breaks down them into maltose. Is it clear? But the medium is alkaline and the enzyme working is amylopsin. So amylopsin acting on starch and complex sugar converts them to maltose. Now the last of these four enzymes was steepsin. It is a pancreatic amylase acting on emulsified fats, converting them to fatty acids and glycerols, though all are happening in alkaline medium. So alkaline medium is the common thing. So alkaline medium is the common thing, and these all are being converted to the end product, that is fatty acids and glycerols, all in alkaline medium. So this is all four enzymes which were present in pancreatic juice and which acted as in enhancing the speed of the reaction, in enhancing the rate of the reaction, and in converting the previous form of the product into the simpler form. Okay, is it clear? Now we will move on to the last juice of small intestine which is being secreted from the small intestine, that is the intestinal juice or the succus entericus. Now here in the intestinal juice, that is succus entericus, it also contains several types of enzymes. Erypsin, which is mainly a group of peptidases. Erypsin is mainly a group of peptidases. Okay, so it contains enzyme erypsin and it is a group of peptidases. It also contains traces of, we can say that it also contains traces of maltase, sucrase, lactase and lipase enzymes. So we will be discussing about all these enzymes right now. Now first we will discuss about erypsin. It is an intestinal peptide. It is what we can say that it is an intestinal protease, that is it is acting on peptides, and end product is amino acids. So it is an intestinal protease. It is a protease enzyme. So as it is a protease enzyme, it is acting on the peptides and end product is amino acid, and the medium always should be alkaline medium. Okay, the medium is alkaline medium and the end product is amino acid. Next comes maltase. That maltase, that is an intestinal amylase which is acting on mainly starch type of substances. In the previous one, if you remember, we have already told that it is acting on the starch like substances. So maltose, it is acting on the maltose and it is converted to much simpler form, that is glucose in alkaline medium. Next, sucrase, another enzyme acting on sucrose, converting to glucose and fructose. So all end products we are saying glucose and fructose. Now lactase, it is converting the lactose to glucose and galactose. And last but not the least is the lipase, which is acting on the emulsified fats and it is converting to fatty acids and glycerols. So all this thing is happening in alkaline medium, and the enzymes generally they are varying. One is peptidase, one is maltase, that is intestinal amylase. Sucrase also amylase. Lactase is acting mainly, it is a sugar lactose, milk sugar lactose, that is converting the lactose to glucose and galactose. And the lipase is, it is actually converting the emulsified fats to fatty acids and glycerol. So by this time we can say that food is completely digested and it is converted into a liquid form, that is called chyle. So first in the stomach it was chyme, now it is chyle, and which is further subjected to the absorption in the small intestine. So this absorption part we are not going to take today, so it will be too much for us, as we have already studied about the enzyme. We have already studied about how the enzymes are converting one form of substance to the end products, how they are acting in alkaline medium. So just go through them thoroughly and remember them and try to memorize them thoroughly, so that when I am going to the next absorption and assimilation part, it will be very much easier for you to understand each and everything very clearly. Now let us move on to the last slide. Now here we will see that how the digestion happens in the large intestine. Practically, no digestion happens in the large intestine. As large intestine, digestion is not taking place. What is happening? The mucus cells secrete mucus, which lubricates the intestinal lining, and it helps the easy passage of undigested food from the stomach to outside. So the food is passing from the stomach to the outside with the help of the mucus, which is being secreted by the intestinal lining, that is the large intestinal lining. So this is what happens in large intestine, but there is no digestion which is happening in large intestine. So is it clear to everyone? I think I have made myself very much clear by taking each and every enzyme separately, by taking each and every juice separately which are being secreted in the small intestine. And I think if I have made myself clear, if you are having any queries, write it. When I come with the videos of assimilation and absorption, I will surely clear your doubts so that you don't have any doubts as in this lesson. If you are having any doubts with my previous lessons also, please write, and don't feel anything to write. Write your queries. Let me understand and clear them up to you. So as of today's topic, it's clear, and I think it is you will be understanding it in a lucid way. So thank you.